So I'd like to begin by thanking Dean Hirsch and the School of Pharmacy and Pharmaceutical Sciences for nominating me as the inaugural recipient of the Athalie Clarke Research Award for our school. It's really just a tremendous honor for me to be recognized by my colleagues for the work that we do here at UCI. I would also like to take this opportunity to thank Mr. James Swinden and other family members across the generations who have supported research through this and other UCI programs. Your generosity is truly appreciated, and we in the faculty are grateful for your high level of community engagement these past many years. Finally, I would like to thank all of the current and past members of my lab. This award, more than anything, recognizes their hard work and their dedication to team science. As Dean Hirsch mentioned, my lab is primarily focused on early stage drug discovery. We ask ourselves two important questions. First, what are the biggest challenges facing drug discovery today? And second, how can we address these challenges? The challenges that we are studying include: one, cost and quality. How can we make better drugs at more affordable prices? Number two, personalized medicine. How can we design drugs that match our individual genomes? And three, targetability. How can we produce drugs against targets that are currently thought to be undruggable? If you look at the top 10 best-selling drugs annually, more than half are human monoclonal antibodies. These are the blockbuster drugs like Humira, which was developed to treat rheumatoid arthritis. Although these drugs are very effective, they're also extremely expensive. A typical drug regimen can cost $80,000 to $200,000 per year per patient. The high cost of these drugs is due primarily to their high cost of discovery and their high cost of manufacturing. These drugs are isolated from animals, humanized, and produced in cells, which is a slow, painstaking process that is not easily scalable. Our solution is to replace monoclonal antibodies with synthetic antibodies that we call aptamers. These are molecules that we can evolve in a test tube and optimize by machine learning. This strategy is expected to provide a cheaper and more available alternative to antibodies by bypassing the need for animal discovery and cell-based production. In the area of personalized medicine, we've developed a promising approach for tackling disease targets that were previously thought to be undruggable. It turns out that throughout our life, from birth until death, our body is constantly producing new cells to replace old cells. During this process, genetic mutations are sometimes introduced into our DNA through mistakes that are made during DNA replication. These are somatic mutations, which means that they can't be passed on to our children, but they can affect our health if they occur in a region of our DNA that is part of a critical biological pathway. A classic example of an undruggable target is a gene called KRAS, which is the master switch responsible for turning cell division on and off. Mutations made to the wrong part of this gene forces cell division to remain on, which causes cells in certain parts of our body to grow and divide in an uncontrolled way. Mutant KRAS genes are found in roughly 20% of all human cancers. In a recent publication, we showed that it's possible to selectively degrade the mutant KRAS gene inside cells without harming the healthy version of the gene. We believe that this discovery could one day improve patient care by providing a precision approach to cancer treatment. Both of the vignettes that I shared with you today, our work on synthetic antibodies and our work on undruggable targets, are examples of translational medicine, where discoveries made in the laboratory are finding their way into the clinic, typically through industry partnerships. I want to emphasize that this journey really has only just begun, and the Athalie Clarke Award will help keep us on the path. I plan to use this award to support students in my lab. Thank you very much.
Well, that's pretty inspiring too. It's sort of like go big or go home. You are really asking the questions at a scale that I'm sure Athalie would appreciate. Congratulations.
Thank you very much.